Imagine you're a detective, right?
Yeah.
And you are tracking this absolute master criminal.
Okay, I'm with you.
But this criminal changes their disguise completely depending on the city they operate in. So in one city, they manifest as a tumor buried deep in the center of the brain. In another, they look like a tumor inside the eye.
Oh, wow.
Yeah, and then in the third city, they appear as a mass in the cerebellum, right at the back of the skull. So on the surface, you're looking at three completely different, totally unrelated suspects.
Right, the locations are entirely different.
Exactly. But when you finally dust the crime scenes, you find the exact same ancient light-sensing fingerprint left behind at all three locations.
It honestly sounds like a biomedical thriller,
Yeah.
but that is the actual biological reality of what researchers have uncovered here. We are looking at tumors that are anatomically isolated from one another, yet they're united by this genetic ghost from our evolutionary past.
It is wild. So welcome to the deep dive. Today we're pulling from this massive stack of recent oncology research and developmental biology notes.
Yeah, there's a lot to get through today.
There really is.
Yeah.
And we are anchoring it all on this groundbreaking 2026 article. It was published in the journal Cancer Cell by Gu Dinis and an international team of researchers. Our mission today is to dissect exactly how these scientists solved a decades-old mystery surrounding some of the most aggressive childhood brain cancers.
And they did it by tracing them back to their exact cellular origins, which is just incredible.
Yes. So to understand the stakes of this research, we really have to ground the conversation in the grim reality of a disease called pineoblastoma, or PB for short.
Right. So pineoblastoma is a clinically aggressive embryonal tumor. It forms in the pineal gland of children, usually kids under the age of 5.
Toddlers, essentially.
Yeah, babies and toddlers. And the prognosis is, it's really grim, even with the absolute most aggressive multimodal therapies available today.
Which means what exactly for a kid that young?
It means subjecting a toddler to intense neurosurgery, radiation, and heavy chemotherapy. And even with all of that, the 5-year survival rate stubbornly hovers around just 15 to 30%.
Oh, man. Okay, let's unpack this because 15 to 30% survival is just a devastating statistic. And part of the struggle here, right, is that scientists have known for years that pineoblastoma isn't just one monolithic disease.
No, not at all. If you look at the molecular profiling, there are distinct subgroups.
Right, like you have PB-mirin-A12, you have PBRB1, which I know is driven by the loss of the retinoblastoma tumor suppressor gene.
Exactly.
And then you have PBMYC or FOXR2, which is driven by the overexpression of those specific oncogenes. So they have completely different genetic drivers and, different clinical outcomes.
Yeah, and that was the huge roadblock. We knew the genetic mutations driving the malignancy, but we lacked the fundamental context. We didn't have the origin story.
The where and the when.
Right. We didn't know the exact cellular mechanism or the specific developmental stage where these tumors were initiating. And if you want to defeat an enemy like this, just knowing the mutation isn't enough. You have to know the exact biological environment where that mutation first took hold.
Which brings us to the methodology in this paper, because the way they hunted down this origin story is, frankly, fascinating. They took 38 human tumor samples. But instead of just grinding up the tumor tissue and looking at the average genetic expression...
Which is what we used to do.
Right, which is like, blending a massive fruit smoothie and then trying to guess the individual ingredients by tasting it. Instead of doing that, they used single-nucleus RNA sequencing, or SNAT RNA-seq.
And we really need to pause and appreciate that, because single-nucleus RNA sequencing is a massive computational and biological undertaking.
Oh, totally. Break down how that actually works for us.
Sure. So the researchers literally isolated the individual nuclei from over 313,000 individual cells across these tumors.
300,000. That's insane.
It is a mountain of data. And by capturing the messenger RNA transcripts inside each individual nucleus, they could read the active gene expression profile of every single cell, one by one.
So they aren't looking at a smoothie anymore.
No, they are looking at the real-time instructional manual that each specific cell is using to function right at that exact moment.
But having a pile of 300,000 instructional manuals doesn't tell you much unless you have something to compare them against, right? You need a reference point to see exactly where things went wrong.
Exactly. And because we are talking about a tumor of the pineal gland, they needed a map of how a healthy pineal gland is built from scratch.
So they made one.
They did. They generated this highly detailed developmental atlas of the mouse pineal gland. And they didn't just look at a fully formed adult gland. They mapped the transcriptome at very specific temporal checkpoints.
Like a timeline.
Yeah, starting from embryonic day 13, which is deep in fetal development, all the way through various stages up to postnatal day 21.
Yeah.
They computationally tracked exactly how the gene expression networks shift and change as a generic stem cell differentiates into a mature pineal gland cell.
I was actually reading through this mapping process, and I started thinking about it like a bustling construction site.
Oh, I like that analogy.
Yeah, so think about it. Like, when you look at a mature pineal gland, that tiny little pine cone-shaped structure deep in the brain that makes melatonin to regulate our sleep, that is the finished building.
Right.
But what the single nucleus data revealed is that the pineoblastoma cells, they don't resemble the finished building at all. They actually resemble the temporary scaffolding phase.
That is a perfect way to visualize it. The human tumor cells, regardless of whether they were driven by an RB1 mutation or a MYC overexpression, they all mapped back to a very specific transient developmental window in that mouse atlas.
They align with a state called pinealocyte progenitors. So these are parent cells that are supposed to differentiate into mature pinealocytes, but they just get stuck.
They get completely frozen in time. So in healthy development, this progenitor state is highly mitotic. The cells are furiously dividing to build the physical tissue mass of the gland.
They're multiplying like crazy.
Exactly. Their gene expression is heavily enriched for programs that drive the cell cycle. But this is also their critical vulnerability. If a cell suffers a major oncogenic hit right then.
Like losing the RB1 tumor suppressor.
Right, which normally acts as the brake pedal for cell division. If it loses that brake pedal while it is in this highly mitotic, vulnerable state, the consequences are catastrophic. The cell cannot exit the cell cycle. It cannot mature.
It just keeps dividing.
It becomes trapped in an infinite loop of replication, and that forms a highly aggressive embryonal tumor.
Okay, so let's just step back for a second and trace the narrative so far. Let's do a quick recap.
Good idea.
So, bullet 1. Scientists have essentially proven that this deadly cancer, across all its various genetic subgroups, originates from a temporary biological scaffolding, these transient pinealocyte progenitors.
Right.
And bullet 2. The pathogenesis is driven by aberrant cell cycle control. They get frozen in a state of rapid division.
Perfectly summarized.
And honestly, for you listening right now, just think about that for a second. Have you ever considered how a tiny glitch in a temporary cellular phase, a phase that only lasts a few days during fetal development, could echo into a devastating disease years later? It's terrifying.
It really highlights how fragile development is.
It does. But as I was poring over the RNA profiles of these tracked progenitor cells, I hit a massive conceptual roadblock in the paper.
Oh, you're talking about the developmental signatures they found active alongside the cell cycle genes.
Yes, because it makes absolutely zero sense anatomically. The data shows that while these cells are trapped in this rapid division phase, they are highly enriched for gene signatures related to eye and retinal development.
I know, it sounds crazy.
Hold on, I have to stop you there because the pineal gland is buried right in the middle of our skull. It is underneath a thick neocortex. It is pitch black in there.
It is very dark, yes.
Its job is to produce melatonin, so we get sleepy at night. There's absolutely no logical reason for a cell deep in the center of the brain to be utilizing a genetic program meant for sensing light. Why on earth are retinal genes showing up in a pineal brain tumor?
It is a massive paradox, and to unravel it, we actually have to look away from human anatomy completely and dive into evolutionary biology.
Okay, take me there.
So in lower vertebrates, think fish, certain amphibians, and reptiles, the pineal gland isn't buried deep in the brain. It sits close to the surface of the skull. It is quite literally a photoreceptive third eye.
Wait, like an actual eye?
An actual eye that contains active photoreceptor cells. It directly senses ambient light to regulate circadian rhythms and seasonal behaviors for the animal.
An actual functioning third eye with photoreceptors, that is wild.
It is. But over millions of years of evolution, as mammals developed a massive neocortex, the pineal gland was pushed deep into the center of the brain. Because it was shrouded in darkness, it obviously lost its direct light-sensing capability.
Right, the light couldn't reach it anymore.
Exactly, had to adapt. So it started relying on neural signals routed from the actual retinas in our normal eyes to know when it's daytime or nighttime. But here's the critical mechanism. Evolutionary biology rarely just deletes old code.
It just silences it.
Or repurposes it, yeah. The ancient genetic programming for phototransduction, for physically sensing light, is still sitting right there in our DNA.
It's like an evolutionary ghost hiding in the genome.
That's a great way to put it. And during human embryonic development, as the pineal gland is forming, that ancient phototransduction program still flickers on for a very brief transient window in those specific pinealocyte progenitors. It is a biological echo of our evolutionary past.
Wow, that completely reframes how we think about this tumor. The pineoblastoma cells aren't actively trying to grow an eye in the middle of the brain. They're just locked in a developmental phase where that ancient evolutionary ghost happens to be awake.
Yes. And the researchers recognized how profound this was. They wanted to know if this evolutionary ghost was present in any other cancers.
So they went hunting.
They did. They aggregated an enormous data set. They pulled in over 90 different central nervous system tumor types across nearly 3,000 human samples.
That is a lot of data to crunch.
It took some very sophisticated computational analyses to isolate the specific genetic fingerprint of this light-sensing program, but they did it. They discovered a highly specific 175 gene network.
And they named it TAPS, right?
Right, TAPS, the Tumor-Associated Photoreceptor Signature.
And the results of that data sweep are staggering. They found that TAPS only unifies 3 anatomically distinct pediatric cancers out of all 90 types.
Only 3.
1, pineoblastoma, located in the center of the brain. 2, retinoblastoma, which forms in the retina of the eye. And 3, Group 3, medulloblastoma, which forms in the cerebellum at the very back of the brain. So you have the eye, the center of the brain, and the back of the brain. 3 completely different locations in the body, utilizing the exact same 175 gene TAPS blueprint.
And just to put that into perspective, the statistical probability of 3 distinct tumor types randomly sharing a 175 gene network is essentially 0.
It doesn't just happen by chance.
Not at all. The researchers ran permutation tests, and the statistical median for a shared gene set among random CNS tumors was just 2 genes. TPS is incredibly specific.
I really want to highlight a clinical vignette from the source material here because it proves just how powerful this shared biological blueprint is over basic anatomy.
Oh, the patient cases.
Yes, they detail 2 specific patient cases. Patient PPT32, a 10-month-old girl, and patient PPT39, a 15-month-old boy. Both of these children presented with tumors physically located in the pineal region of the brain.
Right.
So if an oncologist looks at the standard MRI imaging in the anatomical location, the diagnosis is clear-cut pineoblastoma. But the researchers didn't stop at imaging. They ran DNA methylation arrays on the tumor tissue.
And we should explain what that is, because DNA methylation is a crucial layer of epigenetic regulation.
How does it work?
So for those listening and visualizing this, think of the human genome as a massive library of books containing all the instructions for a cell.
Okay.
Methyl groups act like little chemical padlocks placed on very specific books, preventing the cell from reading them. By mapping exactly where these padlocks are placed across the entire library, scientists can generate an epigenetic barcode.
And that barcode tells you what?
It reveals the true, fundamental identity and developmental lineage of a cell, regardless of where it is physically located in the body.
So it's the absolute truth of what the cell is. And when they scanned the epigenetic barcodes of these 2 pineal region tumors from the toddler patients, the padlocks didn't match the standard pineoblastoma profile.
No, they didn't.
The methylation profile completely 100% overlapped with group 3 medulloblastoma.
Exactly. The tumors were physically growing in the pineal gland, but molecularly and epigenetically, they were completely identical to a tumor that normally forms in the cerebellum.
The biological similarity is so profound that the anatomical zip code of the tumor almost doesn't matter.
It really doesn't.
Okay, let's pause and consolidate the narrative up to this point, around the 10-minute mark here.
Good time for recap.
So, bullet 1. The pineal gland shares an evolutionary link with the retina. Bullet 2. TAPS is a 175 gene signature shared uniquely by 3 distinct cancers: pineoblastoma, retinoblastoma, and group 3 medulloblastoma.
Correct.
And bullet 3. These tumors are epigenetically and transcriptionally similar despite different locations. Which, brings us to the ultimate question for you, the listener, right? If 3 cancers in completely different parts of the brain and eye share the exact same architectural blueprint, could they share the exact same critical weakness?
And that is the transition from biological discovery to therapeutic application. Identifying a shared evolutionary signature is fascinating trivia for a developmental biologist, right?
Sure.
But for an oncologist actually treating a sick child, the immediate question is how to exploit that signature. The researchers hypothesized that TAPS isn't just a byproduct of the tumor's origin. They suspected it was a selective dependency.
Meaning what? Exactly. Like TPS isn't just the paint job on the car, it's the engine keeping the tumor running.
That's a great analogy. The cancer is physically addicted to this photoreceptor program to survive.
How do you even prove that?
Well, to prove a tumor is dependent on a specific gene network, you have to look at the master transcription factors regulating that network.
The boss.
Right. Transcription factors are proteins that bind to specific DNA sequences and command hundreds of other downstream genes to turn on or off. They are the master regulators. The researchers wanted to know what happens if you computationally and biologically delete these master regulators.
Right.
To start, they interrogated DepMap, which is the cancer dependency map.
DepMap is an incredible tool. For those who don't know, it's this massive database where scientists have systematically used CRISPR to knock out every single gene one by one across thousands of different cancer cell lines just to see which gene deletions cause the cancer cells to die.
It's an amazing resource.
Right.
But the DepMap data actually had a major limitation here.
Really? Why?
Because pineoblastoma is such a rare disease, there weren't robust patient-derived cell lines available in the database to run these computational checks. So the researchers had to build the biological models themselves. They utilized genetically engineered mouse models, specifically that PBRB1 model we discussed earlier, and they performed intricate in vivo competition assays.
Okay, let's break down how that assay works, because I read this part, and it's a brilliant piece of experimental design.
It really is.
So, you take the aggressive pineoblastoma tumor cells. Using CRISPR technology, you edit a subset of those cells to completely delete the specific transcription factor you want to test.
Right.
Then you mix those edited cells with regular, unedited tumor cells, and you put them back into the biological environment of the mouse brain. That's the in vivo part.
Exactly. And you tag the edited and unedited cells with different fluorescent markers so you can visually track them over time.
Oh, that's smart.
The logic is so elegant. If the gene you knocked out with CRISPR was truly essential for the tumor's survival, the edited cells will slowly die off or just stop dividing. Over time, they will be completely outcompeted by the regular unedited tumor cells.
It's basically microscopic survival of the fittest.
That is exactly what it is.
And when the researchers systematically knocked out the master transcription factors driving the TAPS program, what happened?
The edited tumor cells collapsed. They couldn't survive without that light-sensing network.
Oh.
But, and this is the absolute holy grail of targeted cancer research, when they knocked out those exact same master regulators in normal, healthy brain cells.
Yeah.
The normal cells were completely fine. The healthy tissue didn't care at all that the genes were missing.
That is the definition of a selective dependency.
It is. The specific master regulators they validated through these CRISPR screens are transcription factors named CRX, OTX2, and NUROD1.
Okay, so I was looking at the names of these three master regulators in the data CRX, OTX2, NUROD1, and I realized their first initials literally spell out C-O-N.
Oh, I hadn't even thought of that.
Yeah, C-O-N, which is incredibly fitting because that is exactly what they are doing. They are con artists.
That's brilliant.
These transcription factors are conning the cell into thinking it's still in that highly mitotic embryonic progenitor state. They are actively forcing the TAPS program to keep running. But you know, the beauty of a con is that it requires the con artist to actively maintain the illusion.
It's exactly.
If you use a targeted therapy to remove the CON, CRX, OTX2, and NUROD1, the illusion just shatters. The tumor cell loses its developmental identity, and it dies.
That is a perfect mnemonic. And the implications of those CON-N transcription factors extend far beyond just pineoblastoma. The researchers took this finding and cross-referenced it with patient-derived cell lines for the other cancers in the TAPS triad.
The eye and the cerebellum tumors.
Right. They proved that CRX, OTX2, and NUROD1 are also absolute dependencies for retinoblastoma and group 3 medulloblastoma. The tumors in the eye and the cerebellum are just as addicted to the con artists as the tumor in the pineal gland.
But importantly, they aren't dependencies for other pediatric brain cancers like ependymoma or high-grade gliomas.
Right. This vulnerability is highly specific to the TAPS triad.
Which means we are looking at a huge paradigm shift in pharmacology. Theoretically, if a pharmaceutical company can develop a small molecule inhibitor or a targeted drug that blocks the CON pathway, say, a drug that successfully neutralizes OTX2, that single drug could potentially treat 3 completely different pediatric cancers.
You wouldn't be prescribing a brain tumor drug or an eye tumor drug. You would be prescribing a TAPS inhibitor.
Right.
We're moving away from the traditional model of categorizing and treating cancers purely by their anatomical zip code in the body.
Right.
Instead, we're moving toward treating them based on their shared developmental circuitry.
Okay, let's do our final quick recap. Bullet 1, TAPS genes aren't just markers, they are essential for tumor survival.
Yep.
Bullet 2, master regulators CRX, OTX2, and UROD1 are validated vulnerabilities across all 3 cancer types. So listener, how incredible is it that the very thing making the tumor unique, this ancient light-sensing program, is also its ultimate Achilles heel?
It's poetic, really.
It fundamentally redefines the battlefield. For decades, when faced with a highly aggressive rare cancer, the instinct in oncology was often brute force.
Unfortunately, yes.
You throw the maximum tolerable dose of chemotherapy and radiation at the anatomical location and just hope it kills the tumor before it kills the healthy tissue. But this deep dive proves why pursuing fundamental biology is so vital.
It changes everything.
By zooming all the way in, by reading the single nucleus barcodes of hundreds of thousands of individual cells, and by looking past the arbitrary boundaries of human anatomy, these scientists found a hidden door. We thought we were tracking 3 different master criminals operating in 3 different cities, but the data proved it was just one evolutionary ghost wearing 3 different masks.
It is a profound synthesis of evolutionary biology, computational genomics, and clinical oncology.
It really is.
We started with a rare, devastating childhood tumor and mapped its origin to a fleeting, highly mitotic progenitor state. We discovered that this state harbors an ancient light-sensing ghost from our evolutionary past that unexpectedly unites tumors in the brain, the cerebellum, and the eye.
So cool.
And finally, we proved that this shared signature isn't just a quirk of developmental biology. It is a shared dependency that could revolutionize targeted therapies for children who desperately need them.
The elegance of the science is just incredible. The very thing making the tumor unique, this ancient photoreceptor program that has absolutely no business being turned on in the pitch black center of the brain, is ultimately the tumor's downfall.
And as we close out this analysis, I want to leave you with a lingering thought about the broader implications of this research.
Oh, lay it on us.
The TAPS signature is essentially an echo, right? A third eye hidden deep within our DNA that gets accidentally switched on during a microscopic developmental glitch. Yeah. It forces us to ask a much larger question. What other ancient evolutionary genetic ghosts are hiding in the human genome, just waiting for the exact right microsecond of cellular confusion to awaken them?
Oh, wow. What else is lurking in the biological scaffolding of human development? Think about that the next time you look up at the night sky. The very light hitting your eye is bouncing off a genetic program millions of years old, one that holds the key to saving lives today.
Until next time, keep digging deeper.